And now we have a very interesting session, the Ask Me Anything panel. So I will hand over to Jay to moderate this, and let's see what this anything can be.
Yeah. I know, they're not here. Can we have the panelists come? And so we have Sarah and Aviv, Musa and Christine. I think Fabian and Dana should also be here. I don't see Dana. Okay. Okay, let me just quickly introduce what this session is all about. It's called Ask Me Anything, and not me, but the panelists. And we kind of started this maybe in the previous meeting in Tokyo. It's one of the ways to really engage with the community because, Sarah and Aviv have been really busy running around and making sure HCA is running properly. And many of you may have some questions that you've been wanting to ask, but they're always talking to somebody and haven't really had a chance to, ask them for any questions. And now here's your chance. So it's a way for you to ask any questions regarding organization to technical aspects to vision and whatnot. So I really want to open the floor up to you. It's ask me anything, but again, don't ask any personal questions, like what they had for breakfast. Don't really care about that, but more like a town hall discussion. You're really here to ask questions. They're not running for president, so it's okay. You can ask any questions you want. But basically, it's an open forum. We also have live streaming where people can also ask through the internet. So really the floor is up to you, and just want to give a lot of these, the panelists a chance to speak. Just quickly, maybe you guys all know what Aviv and Sarah are doing. Christine has been spearheading a lot of GTEx activities, so a lot of experience in tissue procurement. Fabian has been spearheading a lot of bioinformatics and analysis working group together with Dana Pair. And Musa, as you heard today, is part of the equity aspect of the Human Cell Atlas. So you can ask related to these topics or anything outside as well. So floor is yours. So if you can have the lights back on in the audience, so I can see the hands. So anyone? Maybe I'll start as a moderator, because so please think about the question in your head while I'm asking this. So maybe this is more question to Aviv and Sarah in terms of the vision and so on. So the Atlas 1.0, like what is your kind of image or picture of what this Atlas looks like, and how far are we, and what's missing right now, and what do we need to do to really get there? Maybe I'll have this, Sarah.
That's a great question to start off with. Is this actually on? Oh, no, I just press this. Okay.
There you go.
Okay, got it. Yeah, so that's a fantastic first question, and very appropriate. And I think the Atlas 1.0, we laid it out in the white paper, and actually that the vision of having extensive coverage, what was it, hundreds of thousands of cells for a list of priority of the top organs. I mentioned the 5 vital organs and so on in the opening talk, and gave that overview, if you remember, of where we stand in terms of numbers of cells. And then we knew that the spatial, even at that time, when we wrote the paper, all of us together, right? We knew that the spatial technologies were coming, and that the coverage would slowly gain momentum for the organs. We had 12 in the top priority, and then the remaining sort of 40 tissues kind of lower down. I think that's all playing out, and we're actually, everything is happening much faster than I anticipated. And so I think we will reach that version 1.0 vision actually probably within the next 2 years or so, as a community together, that will have coverage of not just even one tissue, one sampling location of an organ, but multiple probably. So I would say we're even doing better than what we laid out in the white paper.
Do you have some, is there a number or is there a size or in terms of data release of 1.0, what would be the requirement, so to speak, to really release that?
So for the DCP, the releases, they're
it's
fast.
Open, of course.
They're fast. It's open, and so they can be
correct, but
incrementally growing all the time.
But in terms of as a community, we're saying this is
when
going to be
when do we make a big announcement?
Exactly. This is, and what does that look like is kind of the question.
I maybe can comment on that. We actually laid out some numbers in the white paper, and some of them we went way overboard already, and some of them were not there yet at all. So I think the area where everyone was really cautious was number of individuals. Probably in 2017, we all felt that getting to 20 people would be ridiculously difficult to achieve goal, and we're now in the triple digits, and it's not even beginning to get to to plateau out. So there it's a little now mismatched, and we need to rethink, and that's part of the roadmap exercise. But I do think that this notion that there is some percent rarity cell for a given tissue type or organ or system that we strive to achieve is probably going to be the main benchmark, and that 1% still comfortable to people, but we might go even lower than that, again, because we're doing better than we expected to be. So it's a little bit of a moving target these days. That was the goal of this roadmap, to make these things very numerical again, as they were in 2017, but the techniques got better. And so we can revise a little bit our ambitions. Having said that, for a 1.0 to be successful, we said those 12 systems that are in the white paper. It's perfectly fine if we got more, but we should at least get those 12. I think in those systems, we should assume we sampled the major anatomical locations, the brain being kind of a special case in its anatomy, but for the others, that tends to have some repetition. So it's not going to give you a whole organ at the end, but it will give you some sampling across the anatomy. Within the anatomy, to have histological, spatially resolved information and depth enough in the cells and their molecules are not only RNA. That also matters. So in the white paper, I think we committed to single-cell RNA-seq and single-cell chromatin for sure, because we knew already we had the scale then, and for spatial methods, for sure, both for RNA and for protein, because we knew the techniques are getting there. If you look at where we are right now, on cells with RNA, we're in terrific shape, and it's clear that the chromatin measurements are also in great shape. They just are lagging temporally behind. But there is no technical barrier to adoption. If you look at the spatial side, I think there's still a barrier to adoption, meaning great people who invented these methods or were early adopters do amazing things. We've seen them on slides. But it's not something that has, everyone does it already. And I'm eager and enthusiastic to see these amazing colleagues from the spatial field doing these great things and coming to our meeting. And I think saying, with increasing levels of confidence, if you just freeze tissues for a bit longer, we'll be ready for them, because that's actually written in the white paper, that we'll be freezing some tissues, assuming these people will come for them at some point. So but that's not for me the Atlas 1.0, that's the data of the Atlas 1.0. Everything I've described so far, the individuals, and there will be metadata, and the anatomical locations, and the histologies, and the molecules that are mapped there in the cells, that's data. That's not an atlas. An atlas has to be derived from the data. So that turns back to what we've described earlier yesterday, which is what are the conceptual entities we want to be derived analytically out of them? And those are, these cell types and the metacells and the states of the cells, the programs of the cells, and how those things map inside spatial organization, so they're associated with histological and anatomical features, and also how the cells then relate to each other in communities, like the ones that Bernd, for example, showed today, and Matt showed the organization spatially as well. So those are the entities that make the atlas. And then we have to have a set of querying tools in 1.0, with that people can come with a query and get answers with respect to those entities. If we had all three things, we had that volume of data, we had at least each of the, and the trajectories, it's all dynamic too. Each of these set of entities and dynamic processes defined based on the data in each of these, and we had a good set of queries, I would say we're in pretty good shape. I'm sure people will actually exceed that. I can imagine many things in the back of my mind that in this rich and amazing community, people will go way beyond. But they might not then do it for all of these tissues and all of these organs, and so they would be the avant-garde that would help us define the next thing. And I'll say one last comment. The white paper was, again, very cautious because of the worry about the number of individuals on human genetic diversity. And I think for some systems where tissues are accessible easily in humans, we will get there for sure. I mean, we saw Shaim already describe that, basically. quickly.
Great.
But of course, for other tissues where there are other challenges, like the magnitude of the tissue is much larger, as in the brain, or the accessibility of the tissue is much lower, as also in the brain, you might not get to the same level of understanding of genetic diversity that we will get to with PBMCs.
Great.
But there might be like some branches in 1.0 that go beyond, and again, show the way for 2.0. Sorry, it was
Great.
a very long answer.
But it was very clear, and I think it was very helpful that you laid out these 3 points. Dana, did you want to add something? I saw you.
No, Aviv mostly said it. I just want to stress that the data will be made available as soon as possible so everyone can use it, and that's a strong philosophy of, making the data as available as possible. But me personally, I won't be comfortable making any large announcement until there's an entity there that I can stand behind that's more than a pile of data.
Yeah. Roderick, is that? Yeah.
Yep. So it's not a question, it's a comment, but it refers to equity. I wanted to make it this morning, but I couldn't. This morning it has been mentioned, the Earth Biogenome Project. So the Earth Biogenome Project and the Human Cell Atlas are the two largest, most ambitious projects in the area of genomics. Now, their scope is very different. Many of the issues related to ethics and equity, I think they are similar. For instance, the consent to get samples from species that are protected or that had particular meaning to local communities, or the limitations to export biological samples outside of the national boundaries, or in the case of the equity, that all geographical areas on the Earth are equally sampled, and that scientists are empowered in the nations where the samplings are being performed and the sequencing, and that this Earth Biogenome Project doesn't really becomes again a form of scientific colonialism or Victorian colonialism, right, in which the white people go to the other places of the Earth. So I think that a dialogue between the two projects, at least in the terms of, I think there are many other issues in which the two projects could establish a dialogue. But I think in terms of equity and ethics, that's actually something that would be really beneficial from the two sides.
Musa, you want to comment on that?
Well, I think there's a lot of really important things that you've said. And I resist the idea of conflating too much ethics and equity. It's a pity that you were not here earlier today. I showed a little cartoon of which I think kind of encapsulates equality versus equity. And this cartoon it's not mine. I didn't make it. It's a cartoon that sort of shows 3 people trying to watch a baseball game behind a fence. And on the left side, the cartoon has a very tall person who can see over the fence and can see the game, and 2 other people who are shorter than this tall person. One of them can barely see over the fence, and the other one is very short and can't see over the fence. That's kind of, they're all equal because they're all equally standing on the same surface, and the height of the fence is the same. On the equity side, you kind of have these different boxes that the 2 shorter people can stand on, so they too can see the baseball game. And I guess from this example, and why I'm being so didactic about it, is that you can see the ethics is not entering this discussion between equity and equality. I think there definitely is a strong component of ethics in terms of how we go into different parts of the world, how we sample populations across geographies, across ethnic groups, across gender. I think definitely there's serious ethical questions and ethical approaches that should be taken. However, equity also refers to the ability to empower people broadly to be able to perform most of these scientific techniques in situ, where they live, and to do them at the same standards that we here are doing them in, let's quote unquote, the developed world. And so I think one of the things we want to do in the HCA, and I think we're striving very hard to do that, is to be equitable in terms of how we build the first draft 1.0 that Viv has come up with. And I think, not to rehash everything I said earlier, but from the highest level of organization of the HCA, I think there's a very firm commitment to have equity in this. I don't know if you want to add anything to that.
I might add one thing. I think, first of all, we're all learning in this. Definitely I am. I told Musa earlier today that I think this is an area where everyone has their heart in the right place and they really want to do well, but they don't really know what to do. And so that's why I said I think for this community it's a learning experience. There are lessons that were learned in how people have addressed this question in human genetics. I think one, and definitely in the Human Genome Project, I think one of the things that, especially in this moment in time, is that it is easier to be equitable from a technical point of view than it used to be in the past. It's easier because our techniques are more deployable, because the cloud exists, because mobile exists, all sorts of things that used to be very centralized in the world are more decentralized today. So the burden of trying to bring things more out from where they were originally initiated, we already benefit from it as a community, and we could now take it responsibly and move it on. And that is less about ethics. That's more about equity. Ethics is as important for HCA, and there's a place where the two meet and where we have to be very mindful and sensitive, for example, in protected populations and indigenous people. But the more equitable you are, I think sometimes it is easier to address the ethics challenge. Because, for example, if work is done in situ, in the originating place, then you're already complying with the ethical standards of that community, where, as opposed to, we're trying to pool the work somewhere else. And I think this will be an evolving item for us, and the more people engage in this, I think the more empowered and knowledgeable we become.
Thank you. Yeah.
I guess I have the mic. Musa, first, thank you to you and your colleagues to taking on such a critical effort. I think it is very commendable. And so your cartoon very elegantly illustrated the concept of equity and empowerment. And so I was wondering, from a practical standpoint, you did illustrate this morning how the community could contribute. But from a practical standpoint, now that you've been to Brazil and planning to go to Ethiopia, like, how are you financing this? Like, how can we help you raise this type of funds? Can some of us help commit like sort of percentage of our grants? Like any comments or suggestions how we can best help the group?
Yeah, we have a collection at the door. So feel free to drop any pennies you have in it. No, I mean, that's a really great question. Make sure they're Euro pennies. No. Yeah, this is a very serious point. I mean, I think this roadshow that we did in Brazil was really was paid for by myself and by Alex directly, Alex Shalik. And I mean, that's not a suitable arrangement. And obviously, we really love the HCA, but paying for it out of our pockets is not the idea. So going forward, I think, I think the OC and other people, other funders in the room who are really committed to the equity in the HCA, they're very targeted ways that they could help. And one of them obviously is, like you said, in these educational activities, training, roadshows like the type we did in Brazil, which completely brought them on the map basically. So I think this is a very serious thing, and the seriousness of the commitment of the HCA will be seen in the amount of money they commit to this, let me tell you.
I will add two things. First, the organizing committee has no money to give, but it can help in raising, as we've done, including for the meeting in Addis, where we found some institutional support, and several of the funders are supporting as well, the Gates, the CZI, the Wellcome Trust, and two institutions, actually, in this case, Reagan and Broad, which are giving money for the meeting because they think equity is important. I will say that aside from money, or directly money, people here have effort, and I think one point that Musa made in his talk was training people well. Could require, people who are in this room to get on a plane and fly somewhere and actually do that training. So you have to pay for the plane ticket, but that's a little different than some of these other things, and we do pay for our own travel for a lot of purposes, but you mostly have to pay with your time and your effort and your attention, and if we can assemble these kinds of teams from this community, we can go very far.
I mean, can I add another comment or request kind of for the equity working group, and that would be, if there are projects, and there will be projects where people will work together, from internationally around the world, like Alex has been doing, you've been doing, and Shyam and Jay actually, and so on. I think what would be useful would be kind of input from you guys on the etiquette, let's say, or lessons learned about what's the best way to go about collaborations and in an equal and equitable way, or whatever, equitable rather than equal. And yeah, to confuse it, you know what I mean. I think that could be it could make things easier, sort of for catalyzing these kinds of things, so that there's also less inhibitions maybe from on both sides.
So that's a couple hands. We'll start from.
So
You had the microphone, right? Okay. Oh, okay, right there in the back.
yeah,
Yes.
so there is a question from Dila Udum from the live stream. She is asking if, do you have any comments on how the work done in the scope of HCA will inform medical education?
Well, I mean, hopefully it will, define sort of physiology version 2.0. I mean, there are already discoveries that will have a big impact on the medical textbooks, right? From new cell types in liver, lung, kidney, brain, you name it. And they'll have, be important for anatomy, physiology, and for pathology and disease. So I think it will have a big impact.
So in addition to discoveries, which are very meaningful, of cell types that play a role in disease, of the structure of normal, and I truly believe that you cannot understand disease without understanding the normal and how it deviates, because that characterizes it. I think one of the things that characterizes this community is that we have accumulated some of the best technical efforts, both in technology and in computation. And this innovation can then later translate, I work both on normal development, also on cancer, and it's a lot of the same computational tools and insights. And I think that this community here has been developing some of the leading technologies, both experimentally and computationally, which have been adopted to medical questions. And many of the people in this community do both at the same time.
Dana also showed a great example in the lung network of how they had to choose how to sample the tissue, and they were definitely motivated by the loci of different diseases. The different diseases tend to occur in different anatomical parts of the tissue, and they try to make sure that in their first pass, they kind of have distinct coverage of each of them. So I think it's not just in the back, it's also in the front of people's minds, while keeping their eye on the goal of building a real healthy reference that then you can use to really understand many different kinds of diseases. Also, in many cases, as we've heard in the gut session, for example, from Gary Betartisian from the Helmsley, there can be, an effort of a normal atlas, and that does not mean that the same people don't also collect data from the irrelevant a disease of interest, and the two basically feed each other. And there are initiatives like Lifetime in the EU that really focus on the disease side, but as Dana said, benefit from the techniques and the computation and integrate. And of course, some of our colleagues participate in both.
Maybe, Christine, if you could briefly mention from your experience with GTEx and also how it's translating for medical purposes and how it could all synergize into a human cell atlas.
Well, what's the specific question with respect to GTEx?
Well, how the data that's been generated is actually being manifested into medical innovation, or at least patient relations and so on. If you can share your...
Yeah, I mean, it's not direct, obviously.
It's not
right?
direct, yeah.
But it was, the design was to understand the sort of functional consequences of disease mutations. So it's taking a step along. Most of our disease-causing mutations are found in GWAS studies, and they appear to act through regulatory mechanisms. And understanding that, it was necessary to look in the different tissues. And clearly, GTEx got us a long way along there. We know a lot of genes and how they and a lot of regulatory SNPs, But it also told us very clearly that we now need to understand that they're very cellular specific, and we need to understand that at this next level. So it's really trying to step towards that functional understanding of how these disease-causing mutations are acting. And there's still extra steps to go, so we're not there, but that understanding is taking us back to really understanding those medical diseases. That's the goal of where we're going.
Yeah, thank you.
So,
okay, go ahead.
Kind of, I guess, to bridge towards that question, how should we be thinking about the best ways of reporting some of our findings in kind of collections of maps that then make up an atlas? And kind of my question is framed in the sense that many of us during efforts to atlas tissues effectively might strike gold in one spot, and so we run back to the journal and we report that we found gold in that location, and then we maybe bury the map in the kind of supplementary data, and then the entire atlas is scattered amongst the different labs that have published those little nuggets of gold in different journals. And so how are we thinking about kind of synthesizing that information together such that we can kind of enable the community to understand rather than just putting the data out there? What other layer of interpretation are we going to add as a community?
Part of the thinking behind that, because it is one of the unique things about the HCA, is it's a distributed effort, which allows everyone and a lot of minds to participate, but it does put a huge burden on the computational people because of harmonization. One thing I can tell everyone that is absolutely necessary to make it possible is very careful recording of how and with what protocols and what details and how that data was collected. That's necessary for any type of harmonization. And that's one of the reasons that I spoke yesterday of adding concepts, because I think having a map where everything will perfectly align at the level of individual cells, taking into account all the differences in the batches and all the cell handling, is going to be very hard. What we can do much easier is really map and connect these meaningful concepts to each other. And so I think version 1.0 was going to be a lot more about directly mapping and finding connections between these concepts, and that connected to the different data sets that brought these concepts through an easy queryable DCP will be the first version of a connection.
I'll give you a couple more things to your to-do list now, checklist, whatever. So first of all, actually deposit your data to the DCP, which would require you, as Bartha described, to have the right consent so that you could deposit it and so on. That's a responsibility, because a journal might not, they would just say put it somewhere. So I think that's important. The second is participate in the annotation process. As you actually were on the path to make your discovery, you noted a lot of things along the way, right? So at least try and complete that piece, even if it's not a big deal for the paper. It's a huge deal for the community, and that goes to Dana's concept. And then, as Peter described the annotation platform, as that would become a reality, and we hope sooner rather than later, participate in that as well. Because these are things where the expert who's seen the data and cares about the data and understands the system, there's today at least no replacement. Maybe they'll train an algorithm, and that would be easier.
I think the key word there was participation. I think that really speaks to everyone to get involved and provide suggestions. So, Sarah, you wanted to say something?
So I guess to make sure that the atlas is transparent. And that all the data, information, and knowledge is encoded. We're working together on achieving that. And I think you heard this morning about the cell annotation efforts. There are other facets that aren't purely annotation, but they are developmental relationships, state relationships, and so on. And I think as a community, the computational group is getting extremely good at encoding those things, visualizing them, allowing them to be queried. We heard the concept of the covariate structure and gene modules and so on, which is another layer. Where what's going to come slowly is how to encode principles of tissue architecture, basically, which is lagging, but it will come, and how to query that and how to recognize computationally microanatomical structures that are teeny tiny relationships between cells that are repeated and so on. And that's, we need to be mindful of those, that level within the larger framework of microanatomy, anatomy, and organs. And so
Just to
on.
comment on that, it's not that we've been ignoring the anatomical level. One of the reasons that we're so mature in the more genomics layer of things is that we have enough data to see the data, to see the signals, to understand the data, to understand the structure of the data so that we can build these concepts. Right now, the spatial technologies are only beginning to emerging. There's not enough data to see the patterns and to see the generalizable concepts that we can build from them. But once that data exists, there is high eagerness in the computational community to extract those concepts from the spatial data as well, and I have no doubt in success given the data.
Right. Maybe related to what the question was also regarding participation, how can we further motivate and incentivize some of the members to partake in a lot of these activities? I know it's maybe a difficult question, but maybe, yeah.
One of the things that I like about the DCP and how it's been organized is that actually it will have a way to quote or to cite the use of data and the use of tools. Because right now you have to pack it all together, package it into a manuscript, and the main citable entity is a manuscript, and people are nervous. Who will scoop me? What will scoop me? Where will my manuscript be published? But if data itself and its use in biological discoveries, and if computational methods that are used to analyze the data and make these discoveries are citable entities that are counted academically, I think that would be a huge incentive to everyone.
There are many things, I think, that incentivize people. So I'm not going to say the obvious ones, like, funding incentivizes people because it lets them do what they want. And so other things that incentivize people is great science, because it makes them, again, let them do what they want. They want to do great science, and when they're around great science, they do better stuff. And so I think one of the things that encourages participation is the open exchange of ideas and the openness to collaboration. So when you go to a meeting, and people don't just talk about the stuff they've done, but they talk about their aspirational thoughts, because that's what they will be doing in a year from now. Not what they're starting to do now, but what they are not even doing, but it's kind of their early concepts and their ideas, because that makes you think better. And if you are open and share in return, then you both think better. And if you also collaborate in the end, because there's something complementary between you, then even more so. And I think that's been the spirit of HCA. That's why people actually drag themselves to meetings, because if the meeting is boring, why would they come? And that becomes this hidden glue. The other really critical thing to encourage participation is to have, I don't want to call it honesty, I'm looking for the right word, is to kind of have fairness in the system. Meaning, if I share my data openly and early, then other people should share then you should share your data openly and early. This was not really me and her,
yeah.
We do. Because otherwise, if there's too much, cheating in the system per game theory, then it disincentivizes people from sharing. But if everyone shares, everyone benefits, and everyone benefits more than, just the sum of the parts. So I think that's a critical thing that will take some time, and the more people share, and the more they see the beautiful stuff that comes from sharing, the more they will share even more. In our community, we already share openly when we actually publish. As we've learned yesterday, that's not a given. But we increasingly share earlier and earlier before we publish. Preprints were not such a big deal 5 years ago. We now all put our papers on the bioarchive, and we don't think that much about it. Data early release was not that common a few years ago. We now do it more. Partly we do it more because funders ask for it more. When funders ask for it across the board, then it's equitable because everyone has to do exactly the same thing. So I think these are the two principles that help. Be open about your ideas and kind of share equally to how other people are sharing with you, and have a little bit of suspend your disbelief, don't assume the worst about people. They end up being actually better than you think.
Great. That's fantastic. Yes. So can you get a microphone in the front? Oh, I think she was first, so. But
She doesn't have a microphone.
all right, go ahead.
I wondered what your current thinking is about how much the human cell atlas should also include data from other species.
I'll start. There's a huge variety of opinions on this answer. Personally, and this is just my own personal thought, I think right now the Human Cell Atlas benefits from more rather than less data, and definitely from the species that are, evolutionarily closer to the human. For example, we've seen how the DCP now made itself mouse compatible, and I imagine there would be a good amount of mouse data, and it's going to be great for the DCP, great for the mouse community, and great for people who chart human cells. I would hope that at least for the major model organisms, we can be hospitable. Now, this is not the same level of commitment as we would have to the human, because it kind of would have to make do with what works for human, and there might not be a lot of specialized stuff that might work for another organism. And I also think it's an open question on which there is a difference of opinion. Mouse is in for now formally. The DCP has already enabled mouse research in this way, but it could be very different for, I don't know, some worms. Although nothing has been said about the worms, just random.
Yeah, so just to be clear, again, following up on that, the data coordination platform, as I'm sure you're aware, is architected in such a way that it can take data from other species. And the nice thing is that there's an alliance that's forming with the fly community now also to use the same architecture. And that's great, because as you showed, like part of the utility of having the human cell atlas isn't kind of to understand our bodies, for medical textbooks, to rewrite anatomy and so on. But another utility is to show what's the value of this model organism for studying this system? What's the value of this model organism for studying this system? How similar is actually, this organ of the mouse to the human and so on? So I think that's going to work out really well and to all of our benefit.
I mean, I'll go one step, and I don't think that we can really do the Human Cell Atlas to a level of understanding, and certainly not a level of, mapping perhaps some lineage relationships without some either organoids or mouse where we can genetically manipulate. And when I ask my research questions, there's always a system where I can do manipulations, which is critical for functional understanding. So I really think that in order to have an understandable human cell atlas rather than what I call a data pile, we will need to incorporate mouse to succeed in the human.
Great. Question one.
Right, we've talked a lot at this meeting about wanting to reach out to other communities, which we all signed up to. But I noticed that, for example, this meeting was oversubscribed incredibly quickly. And so I wonder how, while we all want to be open, there's a danger that we're perceived as actually being quite closed to the outside community because people can maybe not get the access that they might like.
Yeah, I mean, thanks to Helmsley and CZI for supporting the streaming, which is tens of thousands of euros whenever we're here. And that's nowadays one way to make it genuinely globally accessible, also for the future, for all of us to go back to the talks that we heard here, for people who can't travel, for people who are aware of their carbon footprint and so on. So I think that's been amazing at this meeting. I think we started the tradition in Stockholm, didn't we? And then we've maintained it. It's expensive and it requires investment, but it's cheaper than having thousands of people here. It's feasible to have larger meetings. We just need to be clear on how we fund them and whether we charge fees and so on, which we haven't done so far.
So for me, one of the important components of these meetings are the breakouts, and for them to succeed. Part of these meetings, one can, distinguish between informational meetings where, information is relayed, and perhaps we should do more of that, but it's great that this is streamed and the videos are there, and I think they are very widely viewed. But one of the reasons also these meetings are kept smaller, and there is a very varying, if you count over the 8 meetings we've had and get how many names have actually participated, I think half the audience is new each time. But having a small audience actually leads to a successful meeting where there's many more interactions and actually allows whoever does fly over and arrive here to really get involved and give their input. And, we actually do take input from the comments on whoever's watching us online. Yeah. So it's a balance of having something small enough to make progress and to actually communicate and not just have some formal presentations.
So I'll add two things. First of all, this is the first meeting where we did also open registration to anyone who's a member of HCA. It's true that it subscribed so quickly that it actually was not fair because it had to do with a particular time zone. I don't even remember which one, at which time window it happened, but so quickly that some people were asleep. So we're going to change that next time. We already appreciated that fact, and we're going to make like a 24-hour window and a lottery, right? So that everyone gets an equitable opportunity. So there's definitely that. I think travel funds, if we manage to raise them, will improve this as well. But kind of everyone gets penalized at some point on travel costs for these meetings because we may take great care to try and move them around. So, whenever they are in a location, there's also a portion of the meeting that's kind of the local organizer finds local people to engage. So that's another way of increasing this diversity and openness. Nevertheless, it's a challenge for exactly the reason that Dana described. Separately from that, in the AMA that we had in Tokyo, I think Shalin raised the question, or answered the question, I can't remember, related to something similar, to the openness, to access to HCA, how do you participate in HCA? And actually, Shalin and Jay and others have been developing an FAQ that would help people understand all of the ways and mechanisms in which you can engage. And we're pretty close to, not done, but we're getting there. Yeah.
Well, no, it's mainly Shalin's work.
I think Shalin
yeah.
is kind of the lead on that, and
Yes.
He's in Australia right now.
But yeah, I agree, and I think to equity, having these videos and live stream accessible to regions that are very hard to come to these kind of places, it's related to what Musa is doing, and I think that's very critical, and it all ties in together very nicely. That it's not just about having the meeting, but bringing the discussions that we had here to these regions that are not very fortunate to come and travel.
And I think that's been, I mean, it's been covered there, but I think it came out even in the workshop that I attended this afternoon, that there's a great interest in training, hands-on sessions, taking that out. And I think the more that we foster those, I think that's what a lot of the community is really interested in.
Yeah.
So I want to just strongly endorse that, right? And to say what I also said when we raised this in the equity question, this will behoove you people, right, who are the experts. Sometimes you will be the person getting trained, but a lot of the time it will be you going out to train someone else, or the postdoc in your lab, or the grad student. I mean, we have postdocs and grad students in this room, professors and staff scientists and other folks. So that's an investment, but it is so worth it, and it is so much more impactful when you go to someone else and train them rather than when they come to train with you. And we have experience in this. Orit in particular has led an effort like this in the Tumor Atlas Network in trying to have a team that goes around in different hospitals and in different research centers and teaches people certain skills. It's unbelievably impactful, and you might think that you're teaching them because you're the expert and so on. You actually learn a ton when you do that, and your work also improves because they have some tricks up their sleeves too. So it kind of ends up being a lovely way, and people you've worked with side by side, computationally or experimentally, it's a different type of relationship when it goes back to being electronic than when you're just, sending them a file and a protocol.
Well, great. I hope this Ask Me Anything session was useful, and I think it was very fruitful discussion, and I think it was really good to hear their thought processes. So let's give a big round of applause to the panelists.